Assalamualaikum dear students. I hope you're all doing fine. So today we'll be continuing our lecture on carbohydrates that we were studying already. Before we begin with the new topic, today's topic, that's polysaccharides, I'll just give you a quick review of what we had already done, right? So we were studying carbohydrates. Right? And as you all know already, and as the name indicates, that carbohydrates is derived from the word hydrated carbons, right? And it is an organic molecule, a macromolecule. So basically made up of what? Carbon, hydrogen, and oxygen. Three elements making up carbohydrates are carbon, hydrogen, and oxygen, right? And the reason we called carbohydrates was because, or hydrated carbons was, because the ratio of hydrogen and oxygen was same as present in water. That is 2:1. I'll give you a common example that we studied, that is glucose molecule. The glucose molecule's formula is C6H12O6. The ratio of hydrogen and oxygen is same as present in water, that is 2:1, right? Okay, so moving on. Okay, so we studied that there are three classes of carbohydrates, right? The first, or the basic class of carbohydrates that makes up the rest of the carbohydrates, we termed it what? Monosaccharides. They're called monosaccharides. Mono means one, right? So basically a single sugar unit is termed as a monosaccharide. Mono means one, saccharon means sugar. Then we had disaccharides, and we have discussed them also, how they're formed. I'll just give you a quick review. And today we'll be studying polysaccharides. That is the third class of carbohydrates, the polysaccharides, right? Now, as far as monosaccharides were concerned, three examples you studied under the section monosaccharides, and they were what? Glucose, galactose, and fructose. And we studied that all three of them have the same molecular formula, that is C6H12O6. The difference arises in their structural formula. What makes them different is the way their atoms are arranged to form the structure of the molecule. We also studied that the molecule, the smallest molecule of a monosaccharide, is a 3-carbon molecule called a triose. The largest one may be 10 carbon, right? All these are 6 carbon, they're called hexoses. All of these are called hexoses. So these are all hexoses, 3 hexoses that you studied: glucose, galactose, and fructose, right? Okay. Then we discussed that the molecules, when they become larger than 4 carbon, like 3 and 4 carbon, they cannot fold upon themselves. But 5 carbon, 6 carbon, 7 carbon, 8 carbon, the sugar molecules fold upon themselves, giving rise to a ring structure. Meaning that glucose, after forming a ring, would look something like this, right? So this is basically the ring structure of the glucose molecule, right? So this is a monosaccharide. A single ring is termed as a monosaccharide. Then moving on to disaccharides. There were three disaccharides that you studied, namely maltose, lactose, and sucrose. So three disaccharides. Maltose is made up of 2 alpha glucose molecules, right? Alpha glucose plus alpha glucose, that gives rise to what? Maltose. Lactose is formed by the combination of glucose plus galactose. A glucose molecule and a galactose molecule, or a glucose ring and a galactose ring, they combine and they give rise to what? Lactose. And then sucrose, glucose plus fructose, that gives rise to what? Sucrose molecule. And it was alpha glucose and beta fructose, right? That gives rise to what? The sucrose molecule. Okay. The reaction that occurs between these monosaccharides to give rise to disaccharides, that reaction had a name. Because loss of water occurs, because two OH groups they react, the loss of water occurs, we called that the condensation reaction. And then we studied that that condensation reaction is reversible. That is, by the removal of a water molecule, these two molecules can form links, right? And by adding the same water molecule, they can be broken down back to their monosaccharide form. And the reverse reaction of condensation, that is termed as hydrolysis, right? So two kinds of reactions, condensation and hydrolysis. When two monosaccharides are linking together, are joining together, giving rise to a disaccharide or to a polysaccharide, the linking together is termed as what? Condensation reaction, right? And when the molecules are breaking down, when they're breaking down back into their subunits from the ones that they were made, the reaction is termed as hydrolysis, right? Removal of water, condensation; addition of water, hydrolysis. By removing water, we are linking the two molecules. By adding water again, in the presence of an acid or a suitable enzyme, you are going to break them down, right? So I hope it's clear till here. Then today we are going to begin with polysaccharides, right? What are polysaccharides and how do polysaccharides work? How are polysaccharides formed, right? Okay. As you probably already know, poly means many, right? Mono meant one, di meant two, and poly means many, right? Now, many sugar molecules, they link together and they give rise to what? Polysaccharides. More than one, more than two, sorry. So many sugar molecules, when they link together in a similar method, the way disaccharides were formed by the condensation reaction, they give rise to polysaccharides. How this occurs, that we'll see. The first question is why? Why does the body of a plant or of an animal, or why does generally our body need to convert monosaccharides into polysaccharides? Why can we not store glucose in the form of glucose only? Why do we need to spend ATP to convert glucose into polysaccharides and then break it down when it's required again? Like in our case, in our body, what happens? The extra glucose, all the extra glucose is stored in the form of glycogen. The polysaccharide that our body works with is called glycogen, right? So, in the case of animals, in the case of all the animals, the glucose, the spare glucose, the extra glucose is stored in the form of glycogen. And in the case of plants, the glucose is stored in the form of starch. So this is the storage material in the case of plants and animals. So that means I've given you two examples of polysaccharides. Glycogen is the example of polysaccharides. Starch is an example of, and that we will be studying individually. Also, we'll be looking at cellulose, but that we'll talk about later, right? So glucose is converted to glycogen in the case of animal bodies. Glucose is converted into starch in the case of plant bodies. So how does that occur? We'll come to that. The first question is why? Why do we need to convert, or why do plants need to convert glucose to glycogen? Now there are multiple answers or multiple points that I'll be stating. I would suggest write them down somewhere. The first point, glucose is water soluble. Any molecule that is water soluble cannot be stored Because it will keep moving from cell to cell, right? So the first point why we need to convert glucose to glycogen or glucose to starch is, number one, because glucose is water soluble and able to cross selectively permeable membrane. It easily passes through the cell membrane, and whatever easily passes through the membrane, that will not be stored in the cell, that will keep moving from cell to cell depending upon its concentration in the neighboring cells. To store a molecule, you need to make it water insoluble and you need to make it stop, or you need to make the movement stop, right? So glucose molecule, because of its two properties, that because it's water soluble and because it can cross the selectively permeable membrane, it cannot be stored in the form of glucose molecule. It has to be converted either to glycogen or to starch, right? The second point here, now I'm making just a reverse argument of this point, that starch and glycogen are both water insoluble and cannot cross cell membrane. Now, because of these two properties, starch and glycogen are ideal for the storage, right? They're ideal storage molecules. Another point that I would like to add here is, see, if you're trying to fix clothes in cupboard, right, and you're just throwing them inside, right, you won't be able to fix a lot of clothes there. But if you take them all out and you fold them properly and then you place them nicely, you stack them nicely, you will be able to fit more molecules or more clothes in that cupboard. Same is the case here. If you just keep throwing glucose molecule in the cell without stacking them properly, we'll not be able to fit a lot of glucose molecules. But if we stack them properly, so basically starch and glycogen is the stacking of the glucose molecule. We're arranging the glucose molecules in a compact structure. The compact structures being starch and glycogen, right? Also, the last and the most important point is the osmotic pressure of the cell. Now, what do we mean by osmotic pressure? Osmotic pressure, you can understand this in simple words, the tendency of a cell to attract water towards itself or to start osmosis, right? That is termed as osmotic pressure. Now, more the number of solutes, the higher will be the osmotic pressure. If you will have, let's see, if you have 5 gram of sugar here and you have 2 gram of sugar in this cell, right? Definitely the cell with 5 gram sugar will have a higher osmotic pressure because it will attract more water. It will need more water to compensate for more sugar, right? So that means more the quantity of solute, higher will be what? The osmotic pressure. Now, a statement that I'm going to make here, osmotic pressure depends upon the number of molecules, not upon the size of molecule. I'm writing it down. Osmotic pressure depends upon number of molecules, not the size of molecule. Now, why did I use this, or why did I state this? Imagine 1000 glucose molecules. They are going to disturb the osmotic pressure. But let's say these 1000 glucose molecules make 1 starch. 1000 glucose molecules is equal to 1, or let's say 5 starch molecules, right? Now, I said that osmotic pressure depends upon number of molecules, not their size. Starch is a bigger molecule, but the number is different, right? 1000 glucose molecules is equal to, right, is equal to 5 starch molecules. Now, the number of molecules is lessened, right? That means the starch is not going to disturb the osmotic pressure, whereas the glucose molecules are going to disturb the osmotic pressure, right? These are the three basic reasons why we need to convert glucose to glycogen or glucose to starch for storage, right? So in the case of animals, glycogen is made from glucose, and in the case of plants, starch is made from glucose to be stored as a storage molecule. Both the storage molecules are examples of what? Polysaccharides. Now, the next question that arises is, how is starch and how is glycogen made, right? So moving, I hope this is clear to all of you that glucose is water soluble, that's why it's not storable. Starch is water insoluble, impermeable, that's why it's storable. Also, starch does not disturb the osmotic pressure of the cell, while glucose molecules will disturb the osmotic pressure of the cell. Hence, we prefer storing starch or glycogen instead of the glucose molecule, right? Now, I'll begin with the starch. How is starch made now? Now, basically what happens, I hope you all remember the condensation reaction, and if not, we'll just quickly go through it once again. Okay, so H, OH, H, I'm not labeling all of it, just labeling the first and the fourth carbons. So here you have four molecules of alpha glucose, right? They're all what? Alpha glucose. What happens now? Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6 is outside, right? Similarly here, 1, 2, 3, 4, 5, and carbon number 6 is outside the ring. Same here, 1, 2, 3, 4, 5, and carbon number 6 outside the ring. 1, 2, 3, 4, 5, and carbon number 6 outside the ring. Right, so I've made 4 molecules of glucose. What happens now? Carbon number 1 and carbon number 4, because they're adjacent to each other, the reaction occurs between them. OH group reacts with other OH group, or hydroxyl groups, they react. What happens? H from one side, OH from the other side is removed. A water molecule is eliminated. Right? This oxygen that is left, that forms a bridge in between here, just like this. Same happens here. Oxygen here forms a bridge and a water molecule is given off here as well. Same happens here. Oxygen forms a bridge, right? And a water molecule is given off. So when a long chain is established, we say that we're giving rise to what? A starch molecule. But starch is a mixture of 2 types of linkages, right? We call them amylose and aminopectin. We say that starch is a mixture of 2 types of linkages. The 2 linkages have 2 different names. A specific type of linkage is called amylose. The other type of linkage is called amylopectin. Now, what's amylose? When the glucose molecules join together only in a single line, like this, in a single row, a straight file, we call this amylose. This is amylose, right? This may be 50 molecules long, 100 molecules long, right? The linkage has a name. It definitely is a glycosidic bond, but we call it what? Alpha 1 4 glycosidic bond. A little bigger for you to see. Alpha 1 4 glycosidic bond. Alpha because the glucose is alpha, 1 4 because carbon number 1 and carbon number 4 are involved, and glycosidic because it's a linkage between two sugars, that's why a glycosidic bond. So this bond has a name, alpha 1 4 glycosidic bond, right? So when only alpha 1 4 glycosidic bond is present, that part of the starch is termed as amylose. I'm repeating this up. Starch is the storage compound of plants, and starch is a mixture of amylose and amylopectin. Now, what do we mean by amylose and amylopectin? When the glucose molecule links together in a straight line only, forming what? 1 4 carbon 1 4 linkage, or alpha 1 4 glycosidic bond only. There is no branching, right? There is only a single file of the sugar molecules. That is termed as what? Amylose. How is amylopectin formed, or how is amylopectin different? I'm going to make amylopectin over here. See, carbon number 6 is also free, and carbon number 6 has H, and OH with it. So sometimes what happens, bonding starts forming, or bond starts forming between the carbon number 6 and carbon number 1, right? So I'm drawing a sugar molecule here as well. Now, this is carbon number 4, this is carbon number 1, right? What happens sometimes, the bond does not only form between carbon number 1 and carbon number 4, because the OH of carbon number 6 is also free. So sometimes to make more molecules, or to fix more molecules, what happens? Carbon number 1 attacks carbon number 6 as well. So this OH is going to attack this OH and form bond here, right? This is alpha 1 6 glycosidic bond. 1 6, why? Because it's between carbon number 1 and carbon number 6, right? Now, the molecules that will be added to this side now, they will have 1 4 linkage. वो यहाँ पर 1 4 linkage ही बनाएंगे. मतलब सिर्फ पहले मॉलिक्यूल की लिंकेज क्या होती है? 1 6. बाकी सब की continuously क्या होगी? 1 4. Similarly, this branching can occur here, the branch, because carbon number 6 has a free OH. So whenever we need to add or fix a little more glucose molecules, what do? We form bonds between carbon number 1 and carbon number 6 as well. This portion is termed as amylopectin. When branching occurs, that is what? Amylopectin. So as long as the glucose molecule is linked in a straight chain, that's amylose. And then at certain regions, what occurs? Branching occurs, right? 100 molecules, 50 molecules, 30 molecules forming a branch. This region will be called amylopectin. Then again, no branching. So जहां तक branching नहीं है, इसको हम क्या कहेंगे? Amylose. जिस region में starch के branching हो जाएगी, वो region क्या कहलाएगा? Amylopectin. और आगे फिर कोई branching नहीं है, hence again what? Amylose. So what do you call? Starch is a mixture of amylose and amylopectin, right? Amylose contains only 1 4 linkage, whereas amylopectin contains 1 4 as well as 1 6 linkage. Amylose is unbranched, whereas amylopectin is branched, but both of them are present what? Where? In a starch molecule. So a starch molecule is a combination of amylose and amylopectins. I hope this is clear to all of you. And starch is basically the storage molecule of plants. Glycogen is no different. Glycogen looks just like a starch, but the only difference is glycogen has more branching. Glycogen is also made up of glucose molecules, all alpha glucose molecules. The only difference is Glycogen contains more amylopectin-like molecules. It has less amylose-like and more amylopectin-like molecules. That means glycogen has extensive branching, because definitely animals need a lot more energy as compared to plants, because we have to do locomotion, we have to move, right? So a lot more energy is spent in the case of animals. So we need to store more energy as well. To store more energy, we need more branching, right? So glycogen has more amylopectin-like molecules as compared to amylose molecules, right? So I hope this is clear to all of you. Okay, so we have only one thing left, and that's cellulose, but that's going to take some time as well. So inshallah, we'll continue it next time, right? I'm ending the lecture here. Please do read from your book all of this in detail. I'm writing down the page numbers from page number 30 to page number 34. There are 4 pages that you have to go through from page number 30 to page number 34. And all of you who have not joined Google Classrooms, please join Google Classrooms. I'll be sending a worksheet in there tomorrow for you to solve. That will be based on carbohydrates only. All right. See you next time. Allah Hafiz.